today's video we will look at some of the limitations of constraint based approaches, and we will see that most of these limitations arise from the model itself and not the modeling approach as such, and we will see what they are. So, let us study the limitation of constraint based methods, and then in the next classes we will go on to some applications, examples, and how do we model compartmentalized models and so on. So, what are the limitations of constraint based methods? Can you think of some already? So, the first obvious set of limitations are what plagues every type of modeling approach is that you are limited by the data and so on, right. You are limited by your own biochemical knowledge and so on. But the other important aspect here is that you are somehow restricting yourself only to the metabolic lens of viewing the cell. You only view the cell through a metabolic lens. You cannot predict anything regulatory or something like that. And a case in point is that even if you consider FBA, I mean the lacoperon, FBA will make a wrong prediction on lacoperon. So, it will assume that E. coli grows at a higher growth rate utilizing both lactose and glucose simultaneously. Instead of correctly figuring out that FBA will first utilize all the glucose and then switch over to lactose, because there is no regulatory logic that has been put in here. But that is something we will see in a future class as to how we can implement regulatory logic in flux balance methods. So, there are two types of, there are different kinds of prediction errors that can happen. So, you can, let us say this is the model and this is experiment, right. You can have growth, no growth, growth, no growth, right, or rather you can say GG, these two are good, right, true positives and true negatives. The other two are a problem, false positives and false negatives. So, let us first consider the NGG scenario, which means model no growth, experiment there is growth. What could be the reasons underlying this? And let us have the other thing in parallel as well. So, what could be the reasons for this?
Wrong identification of lethals.
Huh?
Wrong identification of lethals.
Yeah. So, that is the issue, right. So, why is the identification of lethals wrong? So, some common reasons could be missing data in the model and objective function and so on, but beyond that what else could be the reasons? When would your model show growth but the experiment show no growth?
There can be genetic differences with the strain we are working on and the model.
So, right, so that could be an important thing, right. So, subtle strain differences. This is still like a gap in the model, but it is really not that obvious, right. You might be working with MG1655 in the lab and K12 in the model and so on, right, E. coli, and that could be give rise to some of these issues. Very good. What else? Yeah, so incorrect medium. Some of these could also go here, right. So, they don't need to be only one way errors, they could cause errors in both places. Incorrect medium. See, maybe you can just think of it in terms of the model. What is the model? Max V by O such that SV equal to 0 and some each of these could be wrong, right. So, you could have some problem with the objective function. Maybe it does not account for an important metabolite that needs to be produced, So, the model seems to grow, whereas in real life it does not grow. Non-metabolic functions, and commonly regulatory effects missing reactions, incorrect gene protein reaction associations, and so on and so forth. Right. So, obviously this is the more common error. This error is a little less common, but that could again arise out of many of these things. So, this can go here, this can go here, this can also come here, this can also come here, this can also come here. Nearly all of these are, many of these are applicable. Right. So, the most common causes lack of information included in the network. For example, certain important pathways not related to metabolism in which the related gene participates may have been ignored. Of course, the objective function may not be defined properly, it does not include an important compound necessary for growth, and gene deletion may lead to the production of a toxic byproduct, right, which accumulates. So, this is something that we do not account for. Right. So, here we can add that. Right.
Lack of information into important pathways not related to metabolism which the related gene participates.
Yeah, so if the enzyme also has some structural role, right. So, when you delete the enzyme, its metabolic reaction is not important, but it destroys cell structure, so the cell dies. And the other issue is certain isozymes are known to be dominant, right. Currently, we basically assign equal weightage, right. In the GPR, we just say A or B. So, if A is there or B is there, we assume the reaction goes on. But what happens if for A, the reaction upper bound is 100%, whereas for B, the reaction upper bound is 40%. If one gene is like weaker at doing the same job. So, this may not sufficiently rescue the growth. So, when you delete A, there is no effect, or like when you delete B, there is not much effect, but when you delete A, the cell can only grow at 40% of its maximum or something like that. The lack of regulatory information is also a major drawback, right. And usually incorrect predictions are due to false predictions of lethality. This can also be because of other unidentified enzymes. Again, you know, some we did not exactly say incomplete model. So, we could say incomplete model, right. You could have incomplete models or GPRs like, you know, alternate isozymes, etc. So, and by and large, the important thing to note of course is that the inaccuracies stem from problems with the model, not the simulation or the modeling approach itself. Right. So, FBA as such is very reasonable except for the fact that it ignores regulation or, you know, in some sense ignores non-metabolic activities of the cell, although some of it is somewhat accounted for by the ATP maintenance flux and so on. But the flux balance constraint based modeling method by itself is not very flawed. There are, you know, very good agreements with what you find in literature and so on. In today's video, I hope I introduced you to some of the limitations of constraint based approaches. As we have discussed even in a previous lecture, all models are wrong, so are constraint based models, but they are very useful to give very interesting insights. So, and we also I hope you are able to appreciate the fact that the modeling methodology is actually quite good, but you can have a lot of knowledge gaps which affect the accuracy of your predictions and so on. In the next video, we will do a lab session wherein we perform gene deletions using the Cobra toolbox. I will also show you this interesting tool called Escher, and we will also look at how you do single or double gene deletions or synthetic lethals using the Cobra toolbox.